Why to study glycosylation on viral surface antigens? So glycosylation on the viral surface also helps the virus to escape the host immune system by acting as a protective covering against host antibodies and by preventing the adaptive immune responses from host. Glycosylation on the proteins can happen at different locations on the viral proteins, and mutations at these glycosylation sites or different locations on the viral proteins can lead to changes in viral infectivity through altered binding affinity and immune evasion. So sometimes mutations can happen on viral surface protein such as spike protein, as happened with SARS-CoV-2 virus. Several mutations happened, and earlier it was a wild type, then changed to alpha, then beta, gamma, delta, and now Omicron variant. All these variants of SARS-CoV-2 had differences in their spike protein because of mutations. So these mutations led to changes in the structure of proteins and these properties, as well as the changes in glycosylation sites. But interestingly, all the five earlier variants other than Omicron had similar or almost same glycosylation sites. There was no variation in the glycosylation site. Only the variation happened in the case of Omicron variant. So virus attaches to the cell surface of host through glycans, and then later those viral particles are endocytosis sized inside the cells, and then the viral genome will be released into the host cells, and then that will help in further viral replication. That is what shown in this figure. Vaccinations. So vaccines are agents that helps the host immune system to recognize and destroy any microorganisms by training our immune system about the structural characteristics of the microorganisms. Basically, we are using a portion of these microorganisms, either the cell surface proteins or any highly expressed proteins on the microorganisms, and then we use that to trigger our immune system to respond for any future attack of similar microorganisms. There are different types of vaccines, such as inactivated vaccines or attenuated vaccine, or Covaxin is an example for this type of vaccine. Toxoids, subunits, conjugates, or heterotypic vaccines. So there are several new generation vaccines, such as DNA vaccine from the vaccine developed by Zydus, ZyCoV-D is a DNA vaccine. Then recombinant vector, the Covishield vaccines are coming under this class. Then mRNA vaccines, the vaccines developed by Pfizer, Moderna, etc. comes under this vaccine category. So these glycans on the surface of viruses can complicate this vaccine development, because depending on how these vaccines are produced, the glycans can change, and that can affect the subsequent vaccine-based immune system stimulation. So glycosylation is important to understand for the vaccine development. So now we understood the need for the development of glycoproteomics or glycan analysis for the development of therapeutics against viruses. There are several glycoproteins and structural proteins or structural proteins on SARS-CoV-2 virus, such as spike protein, which is on the surface of the virus, envelope protein, which is also on the surface but embedded inside the membrane, and membrane protein. Nucleocapsid protein, which is another important structural protein of SARS-CoV-2 virus, is a phosphoprotein, and that sits inside the cells, inside the virus particles. And that is not a glycosylated protein, it is a phosphorylated protein that entangle the genetic material of viruses. And viruses, as shown in this figure, enters the host cells by uh, the binding of their spike protein with their receptors. So now focus on spike protein of SARS-CoV-2. So this is one of the most important protein for the viruses, since they provide access to the host binding and host entry. So there are two subunits for this spike protein, subunit 1 and subunit 2. So the subunit 1 have the receptor binding domain. So those receptor binding domain, as shown here in the figure, highlighted region, that region will bind with the host receptor. Mostly in the case of humans, those are angiotensin converting enzyme. So once this receptor binding domain or RBD domain binds with the host receptor, there is a cleavage site between subunit 1 and subunit 2. Those are also called furin cleavage site. So this site will be cleaved by the host proteases on their host cell surfaces, and that will facilitate the entrance of the viral particles to the host. So there are 3 units of spike protein attached together in each of the viral spikes, and each spike protein unit has 22 N-glycosylation sites. So altogether there are 66 N-glycosylation sites are present on each spike of SARS-CoV-2, as shown here in this figure. So earlier these protein structures were deduced or understood by cryo-electron microscopy, and these glycans are studied in detail by mass spectrometry, particularly glycoproteomics method. So I'm discussing in more detail about the procedure that we conducted for the glycoproteomics.